So for the last 2 days, last day and today, you've heard a lot about people talking about their tumors, about anxiety, about scans, and about the various treatments. And we're going to take a different step. As I said, I'm Italian. I'm always saying, what can I do? What can I cook for you? And this is a really good session from a caregiver perspective because we all want to know what to do to help our loved ones going through the journey and to give everyone the best shot. So now we're really going to focus a little bit more on the microscopic level. There's a lot of research being done here in Ohio, especially by some of the scientists at the Cleveland Clinic. Dr. Laura Bucavina received one of our Young Investigator Awards and is doing an enormous amount of work on the gut microbiome, how it impacts your tumor, how it impacts how your treatment works for you. And so today we're going to be talking about, from gut health to bladder health, the microbiome connection. And it's a really fast-moving field. There's so much that we're learning today that we didn't know 5 years ago. If Beacon knew 20 years ago, we'll be talking about the gut microbiome, we'd say, what is that? And now today, this is where we're going, to that actual level of literally you are what you eat, and what you eat, food can be medicine, and we need to think about that. So today, I'm really delighted to welcome Dr. Jason Scovell. He's the chief resident in urology at the Cleveland Clinic, Glickman Urologic and Kidney Institute, soon to be the Society of Urologic Oncology Fellow, which is a really impressive characterization. Thank you for all the work that you're doing for patients. With both an MD and a PhD in translational biology and molecular medicine, Dr. Scovell brings the world of science and clinical care together, advancing surgical innovation and laboratory science to help improve outcomes for you all. I think that's really important. So please welcome Dr. Scovell.
Well, thanks for having me here. It really is my honor and my privilege to spend this time with you all. I was able to see that last session, and it's just really a powerful experience seeing you all coming together for one another, and it's just something that we don't always get to see from the clinic perspective. So thank you for sharing that with me. So who here, I'm going to talk about the microbiome as it pertains to bladder cancer, and we'll talk a little bit outside of bladder cancer as well. Who here? Oh, this is the most comfortable talk I think I'll ever give. Who here has heard about the microbiome? Just a show of hands. Oh, okay. So I think a lot of us have heard about it. Who here knows a lot about the microbiome? We've got one expert. We'll bring him up. So the microbiome is a field that's really been developing over the last couple of decades as we've started to understand its importance in overall health, not just the gut, not just any one organ system, but across our entire body. And so I will go over a little bit of bladder cancer, although I think you guys are really the experts here in this. And then delve into the microbiome as it pertains to this disease. So unfortunately, we see about 80,000 new cancer cases for bladder cancer every single year. And unfortunately, this affects both men and women, and the prevalence increases through each sort of decade of life. And I'm not going to get into this, but I think, as you all know, there's different stages of bladder cancer, and based on these stages, it really drives the kind of treatments that we offer patients. And I know there was a session yesterday on sort of what the current state of affairs is, and I know there's another session right after lunch talking about what's coming on the horizon for this. Bladder cancer is diagnosed by direct visualization and then resection of the tumor. And these are typically what tumors look like when we go to the operating room and resect these in the operating room. So what is the microbiome? The microbiome is a community of organisms, and this could be bacteria, viruses, fungi that exist in a particular environment. Now, most of the microbiome studies that are out there today are focused primarily on bacteria, which there's a diverse set of bacteria that live in all aspects of our bodies. So we think about our skin has a lot of different bacteria that live there. Certainly your gut, I think we're all familiar with that aspect of life. But you have bacteria in all aspects of your body. And so what does that matter? You know, are these bacteria just living on us, living in us, but not really contributing to our overall health? I think if you dial back 20 years ago, that would have been sort of the prevailing thought. But what we're coming to understand is that these bacteria and other organisms really live in symbiosis with us. They need an environment for them to live and replicate, but they also provide signals and metabolites that impact how our cells actually function. And so these bacteria can metabolize nutrients, produce vitamins, and they can actually impact how our immune system responds based on which bacteria you have, how many bacteria you have of that type. And so when we think about the gut microbiome, we have trillions of different bacteria within our gut. They can synthesize metabolites, hormones, neurotransmitters. But I think importantly, as it pertains to disease, they can also participate in the metabolism of drugs. So we all think that there's a certain dosage of a medication that we should all take. But based on your microbiome, based on the bacteria that you have in your gut, that bacteria may be more highly metabolized than in somebody else's gut. And that's something that we don't really understand today, is the interplay between one person's microbiome and another person's microbiome. Everybody generally gets the same dosage of drugs. And so in this one study, they looked at 271 different drugs given to patients, and they found that gut microorganisms could metabolize 176 of them. So the vast majority of the drugs that we prescribe to people, they can be altered not just by your body, but by the bacteria that live within your body. And so this is a really important aspect that I think we don't really have a good handle on today, is how do we evaluate on an individual basis. You know, we talk about individualized medicine, but this is, I think, a lot more complex than we all really understood 10, 20 years ago. And so this is one paper that looked at a couple of different drugs and looked at the metabolism of these drugs based on the microbiome, and they found that actually 63 to 78% of that metabolism was due to the microbiome and not the patient themselves. And so this really shows us that this is a big contributor to the kind of activity that goes on within us that doesn't really relate to just us. So how do we study the microbiome? The microbiome is very complex. There's lots of different organisms, and you can't just take some stool or urine and try to culture what is growing in that microbiome. You know, those settings are really ideal for growing a couple of different types of bacteria out, but there's such a diverse array of bacteria that grow within us in these different sites in our body, we have to take a different approach. And so the way that this is often done is that a source, stool, urine, skin, skin culture, etc., is taken and then sequenced. And so they take the DNA, the RNA that they get from that sample, and it is sequenced. And then with computing power, what happens is you're able to take all those little bits of sequence and map them to all the bacteria, and you can sort of look back and figure out what was actually in there. So it's sort of like taking a bunch of different puzzles, mixing them all together, throwing them on the ground, and then sort of going through and trying to figure out this piece belongs to this puzzle, this piece belongs to that puzzle, and figuring out how much of each puzzle you have mixed into that sample. But that comes with some challenges. Whenever we look at the microbiomes across these different samples, there's always the possibility of cross-contamination. We're not looking at millions of each type of bacteria. There's little bits of many, many, many different types of bacteria. And so when we talk about the urinary microbiome, you could get cross-contamination in your samples from avoided urine. There may be cross-contamination from the skin around your urethra, and same with a catheter, and same with other collection methods. So that's all just to say that this is a very challenging area of medicine to study, because the sample sizes are very small, the diversity in the samples are very large, and there's always the possibility that you think you're getting a sample from one place, but you're actually studying some microbiome from other places in the body as well. And this is just to give you sort of an idea of the complexity that come out of these type of evaluations. So this is from a gut microbiome study, and you can see each different little color is a type of bacteria. So, for example, on this list somewhere is Staphylococcus. I think we've all heard of staph infections, and staph is a bacteria, but there's even beyond that, there's different forms of Staphylococcus, and so the diversity here is just a high-level view, but really what you get out of these analyses are many, many different types of bacteria. So urine is sterile, right? I think we've all heard that urine is supposed to be sterile, and we've heard about urinary tract infections, and that is an instance where you don't have a sterile set of urine. But it turns out that urine is not sterile. Urine has its own microbiome, and the diversity within that microbiome is very rich. And back in 2014, really, I think this discovery was kind of very enlightening to the field. I think you look back 20 years ago, we would have all said that urine is sterile. But this group was able to prove that urine has its own microbiome that's very diverse. And actually, in asymptomatic people that don't have anything going on with their urinary tract, they were able to grow out various bacteria, aided by the fact that they were able to pick these up on genetic sequencing.
Can I just want to say
Of
one
course.
thing? You know, Beacon has been at the forefront of research, and Dr. Alan Wolf is the author of the study that Dr. Scovell is mentioning, and he presented back then at a Beacon think tank. And I remember being in the room when there was a collective gasp from 300 clinician researchers who had never heard before that urine is not sterile. The air went out of the room for a second. It was amazing, but that was back in 2012, and you know, that was the most amazing game changer, and now we're having a whole field of science devoted to that because somebody thought to think outside the box, which was the whole premise of our think tank.
No, sorry at all. Thank you, Stephanie. I mean, I wasn't there for that, and so kind of hearing that.
Still in high school.
I wish. But really, I think that story highlights where we are today and where we're ultimately going. This is only 10 years ago that we even realized that urine is not sterile at all. So I would say that our lack of understanding of how all of this works, we shouldn't worry about that. I think that we should be excited of really what's to come. So how does this all work? You know, we think that, again, we talked about the dysbiosis and symbiosis, and dysbiosis being that there's a imbalance in the signals that keep the body healthy. And we're finding out that this dysbiosis can play a role in disease across the entire spectrum: heart disease, inflammatory bowel disease, liver disease, really across the spectrum. And so while I think that there's a lot of work being done in bladder cancer for the microbiome, there's work that's being done across the world, across many, many disease states. This is highlighting that there are thousands of clinical trials currently ongoing across many disease states in the realm of in the microbiome space. And so what we can be hopeful for is that as people make progress in other diseases, that will help us make progress in bladder cancer as well. It's complex, though. You know, when we talk about the gut microbiome, we talked about how this impacts metabolism of drugs and may impact our bladder, impact our urinary microbiome, impact our disease. But it's not just a localized, what is my urinary microbiome, what is my gut microbiome, but there's an interplay, as we're learning, across not just these two organs, but the bacteria that live within these organs as well. So how does the microbiome impact bladder cancer? And I'm going to touch on how does it impact the development of bladder cancer, and then I think also importantly to a lot of you in this room, how may it impact treatment response? So as it relates to the development of bladder cancer, the data is really mixed at this point in time. There are some studies that suggest that there is a difference in the microbiome of people that ultimately develop bladder cancer compared to people that don't develop bladder cancer. We do know that there is a correlation between chronic inflammation due to bacteria, either bacteria that are in a diverticulum or in schistosoma, which is a parasitic infection into the bladder. But this is really with a squamous cell bladder cancer and less with the more common urothelial type. I think the most promising evidence in this space that the microbiome can lead to development of bladder cancer or change whether or not somebody develops bladder cancer. There's some mouse data where they were able to control some of the variables, and I'll show you that data as well. So, as I mentioned, some studies say there is no association between the urinary and the gut microbiome as it pertains to the development of bladder cancer. And there are some studies that say yes. And I think this leads to some confusion about, is this actually a driving factor? And I think what we have to go back and understand is that collecting these microbiome specimens is very complicated, and there's a lot of diversity. And so I think there's still, in this space, an unanswered question whether or not the microbiome can protect you or drive the development of bladder cancer. This is probably the most promising data. This was published in 2024, so just last year in Nature, which is a premier journal, that showed that the microbiome in a mouse model could drive the development of bladder cancer. And so what they did here was they took mice, just standard laboratory mice. That's what those letters on the left-hand side, those are just basic laboratory mice. And they gave them a toxin called BBN. And this is a well-established model in bladder cancer in mouse, like a well-established mouse model, where you give these mice this chemical BBN. It's a carcinogen after it becomes converted into another molecule, which I'll show in a second. And what they did was they gave one group this carcinogen, and then they gave another group this carcinogen plus antibiotics, and they asked, what happens? And so what they found, if you look on the far left-hand side, I know this is a little bit complicated, but I'll just walk you through it. On the far left panel, you see there's two groups, the BBN group and the ABX, which is the antibiotics, ABX-BBN group. And they have different color codings, and what they show is the lightest color coding is no cancer, and the other colors are cancer, but different types. So carcinoma in situ, invasive cancer, sarcomatoid carcinoma. And so what you see is that the BBN group, the carcinogen group, has a lot more of that darker shade color, indicating that those mice that got the carcinogen developed bladder cancer. In the antibiotics, BBN group, you see a lot more of that light shaded color, which suggests that they did not get bladder cancer. And so in this group, for whatever reason, which we'll go into, they did not, the antibiotics were actually protective of developing bladder cancer. So the question is it really the antibiotics that are protective? Probably not. The hypothesis that they were working off here is that the antibiotics were a drug, a mechanism to alter the microbiome in these mice, because these antibiotics could wipe out a whole spectrum of bugs that live within the gut in these mice that, it's not a targeted thing. It's not one particular type of bug, but it's altering the overall microbiome. And so what they found was that by altering the microbiome, that carcinogen was converted at a much lower rate to the actual active form that was driving bladder cancer in these mice. So when we go back to what we were talking about earlier, how our microbiome within our gut contributes a lot to metabolism and metabolism of drugs that are prescribed to patients, it also, this paper suggests, can play a role in metabolizing carcinogens that ultimately our bladders sees and is impacted by. And so this is really the strongest evidence, I think, to date that the microbiome in our gut can impact whether or not somebody will develop a bladder cancer, and that's by altering carcinogens that are seen in our guts. Let's see here. So let's talk a little.
Is it a quick question? Come on. So, I got to speak loudly until the microphone gets here. It's coming.
Thank you. So I worked in the GI space as a nurse practitioner and did a lot of work with inflammatory bowel disease and FMT, fecal microbial transplant. So I'm wondering if there is some type of correlation where you could actually take good, healthy people's bladder and urethral microbiota and transplant. Has there been any work done there?
I think that's a wonderful question and a great segue into our next segment. We'll talk about that some more. That's a wonderful question. So the question essentially was that there's such a thing as fecal microbiota transplant, where you take stool from a healthy patient and give it to somebody who has a disease. And we'll get into some of that here just in a second. So I think that's a wonderful question. So let's talk about treatment response, because I think that's certainly a topic of interest, I think, to the folks in this room. So this group here, what they did was they looked at patients that have cancer before treatment with BCG, and they looked at their urinary microbiome before and after treatment with BCG. They also looked at some microbiome of patients or people that were not patients and did not have bladder cancer as well. And what you see here, this is a lot of colors, but you can see on the far left side, these are people that do not have bladder cancer, and the colors are the different types of bacteria that they have within their bladder. And you can see this is fairly homogeneous, fairly similar across the various patients, so each row is a different patient. And then in the middle column, these are patients that have bladder cancer that were planned to undergo BCG, and this is their mixture of microbiome. And so you can see here, this study would suggest that people that do have bladder cancer do have a different microbiome than people that don't have bladder cancer. But you can see it's still fairly homogeneous. But after BCG therapy, the microbiome in these patients gets completely altered, completely changed. And these are actually matched for the pre-CG and the post-BCG. So you can see even within individual people, that color shift is a complete shift in the type of bacteria that live within their bladders. They were also able to find that the metabolites produced by the bacteria in the bladders of these patients were different among people that responded to BCG and did not respond to BCG. And so this may be another avenue, I think, as you'll hear in the next topic, in the next presentation, that BCG response and unresponsiveness is a big question in the field right now, because BCG is such a wonderful treatment, but not everybody responds to this therapy. And so trying to identify who will respond to this may be somewhat influenced by the microbiome, and that may be something that ultimately we're able to use as a predictive tool. And perhaps way down the road, we may be able to alter, and that by that virtue, alter someone's responsiveness to BCG. When we talk about antibiotics, I don't want to pin antibiotics as a good thing or a bad thing. It's a tool in a specific space. As we saw in that mouse model, the antibiotics actually were protective of the development of bladder cancer. But I'll show you in a couple of additional sets of data here, antibiotics can also be harmful for therapy. This group looked at their impact on BCG treatment, and it did not appear that antibiotics around the time of BCG treatment impacted whether or not patients would go on to recur. This group, Dr. Bukovina, who's also at Cleveland Clinic, and I know has been very involved with BCAN, they wanted to ask the question of whether or not there was a difference in the microbiome to people that responded to chemotherapy. So these are patients that have progressed further than patients that would just receive BCG. These are patients that have muscle invasive disease. They looked at 152 different patients, and they wanted to characterize the differences and try to figure out if there may be a difference for people that responded to therapy or did not respond to therapy. In this study, they did not find any difference in people that responded to chemotherapy compared to people that did respond to chemotherapy. But here, they do find a difference in the microbiome in the urine of people that do have bladder cancer compared to those that do not have bladder cancer. So this is yet another study supporting that there is a difference in the urinary microbiome for people that develop bladder cancer compared to healthy controls. So do antibiotics impact treatment response in cancer? There is some data that unfortunately shows some negative responses. So this is in patients that have metastatic pancreatic cancer, which I know is not bladder cancer. But I think the key finding here is that the drug that they found that was impacted by antibiotics was gemcitabine, which I think some of you in this room are familiar with, is a drug that's commonly used in treatment of bladder cancer. And so there is some signal here that the microbiome may impact the metabolism and the efficacy of gemcitabine. This is some work in the lab that furthers this finding. And so what they did here was they took a mouse model for pancreatic cancer, and in this cancer they injected different bacteria. And what they found that certain bacteria that were injected into the tumor were able to metabolize the chemotherapy gemcitabine and inactivate gemcitabine, as a result, making it less effective altogether. And so when they reversed this effect by treating those bacteria within the tumor with an antibiotic, they were able to restore sensitivity to gemcitabine in this model. And so this, I think, is another set of examples that there's a direct interplay in the bacteria that live within us and live here within this tumor to impact the efficacy of the treatments that we prescribe. Immunotherapy is another area that there has been actually a lot of recent data evaluating the effect of antibiotics, and I think by that token, it's really the impact on the microbiome, but the effect of antibiotics on immunotherapy response. And so this one study published last year, this is all very sort of recent stuff that we're learning about. They looked at 5,000 patients that had urothelial cell carcinoma. I believe this was both upper tract and bladder cancer in patients that were treated with an immunotherapy. They found that patients that had an antibiotic prescribed around the time of this immunotherapy were more likely to progress. But I want to caution you, this is not causation. This does not say that antibiotics will actually lead to you having a worse response. This is not a clinical trial. I think that's really, really important to not leave here thinking that you should not take an antibiotic if you need an antibiotic. But this is some evidence to say that we need to keep looking at this, and we need to understand how does this interplay relate, and then how can we use that interplay to our benefit. Unfortunately, this finding of antibiotic impacting immunotherapy has been shown in other types of cancer, melanoma, non-small cell lung cancer, kidney cancer, etc. So there is some mounting evidence to show that immunotherapy is impacted by our microbiome in some form or another. And I think that's not surprising, as we saw in a much earlier slide, that there is this idea of symbiosis and dysbiosis, where the microbiome can train and impact the function of our immune cells. Altering that may also alter whether or not a drug is effective, less effective, or more effective. So I think to get to our gentleman's question, can and should I change my microbiome? Most of the data in this space is in the fecal microbiome. The urinary microbiome is less understood, but I think for the purpose of this talk, what's really less important is it the urinary microbiome or the fecal microbiome? We have lots of microbiomes, and we need to understand how each of those works and interplay in this disease. But I do want to say that it is unclear in bladder cancer currently whether or not altering the microbiome is something worthwhile. So how do you go about altering the microbiome? There are a bunch of different ways, and we don't have a strong, because the microbiome is very complex. It's not one bacteria or another bacteria, but actually many, many, many different types of bacteria. Altering it becomes a lot more challenging. Different strategies to alter this can be dietary, changing what we eat, prebiotics, probiotics. You can do a microbiome transplant by taking stool from one person and putting it in another person, and there's actually FDA-approved treatments for this. And then there are some other methods by taking engineered bacteria, if we can figure out which bacteria are the ones actually driving these types of events. Really, the early data for the fecal transplant, if anyone here has heard of the fecal transplant, has been in an infection called C. diff, which I'm sure many of you have heard about. And so the idea here is that C. diff is a bacteria that when the microbiome is altered by antibiotics and you lose that diversity within your gut, that typical microbiome that you have, C. diff is a bug that is often not killed by our typical antibiotics that we give. And so it's given the opportunity to outgrow and outcompete all of the other bugs that are within our colon, and it can be a really nasty infection that can cause a lot of problems. Now, the way that we treat C. diff is often with one of a couple of different drugs. One of them is vancomycin. Another one is I'm going to butcher that, fidaxomicin. Those are really our two mainstay treatments for patients that have C. diff. There's another drug as well. But they looked at patients here that had recurrent C. diff, difficult to treat C. diff, and they treated them with vancomycin, which is a very standard therapy in this scenario. They treated them with fidaxomicin, which is a really aggressive drug that can be really effective in this space. And they also tried treating patients with the fecal transplant. And what they found here, if you look at the bar chart on the far left, the dark blue color is the fecal transplant. And this is the resolution rate in this difficult to treat patient population. It far exceeded the ability of our drug therapy to treat these patients and clear them of this disease. And so this shows here that altering someone's microbiome, at least in one area, can impact the ultimate outcome of someone's disease. But what about other diseases? If we're talking about that the microbiome can impact disease, can create disease, can make disease worse, what about a fecal transplant in patients that have other disease states? Can it improve that? And there's emerging evidence over the last decade that fecal transplants may be effective in improving insulin sensitivity, so people that have diabetes, improving chemotherapy resistance in colorectal cancer, drug sensitivity, and various other forms of disease. I apologize, this is not the best photo here, but this is a really powerful study where they looked at patients that received a fecal transplant in advanced melanoma. These were patients that had immunotherapy, did not respond to immunotherapy, and they took a fecal transplant from 2 different donors and transplanted this into people that were non-responders for immunotherapy in advanced melanoma. And what you can see here is that patients that received the red group, so that particular donor, many of them turned into immunotherapy responders. And the size on that graph is the size of their tumor over this time period. And you can see that 3 out of the 4 patients that received from donor 1 became responders, and their tumors shrunk by altering their microbiome in their gut. And I think what's even more powerful here is that if you look at the blue color group, so this is a microbiome from another donor, these patients unfortunately did not convert into responders. And so that suggests that there is something special, something inherent within one microbiome that can impact disease. The question is, what is it? I think the most accessible thing to a lot of us here are prebiotics, probiotics. Do I need to go and buy a supplement today? Do I need to go and start taking these things? And all I can add here is that there really are no FDA-approved products in this space. These are dietary supplements that are, you don't really know what's in them, but more importantly, we don't know, as it was highlighted really in this slide, we don't know which microbiome is going to be impactful in changing your health and changing your therapy outcomes. And so really at this time, I have no current recommendations as to go out and buy an additional supplement of this type. But I do want to leave you, and this is my last slide, is that there are ongoing trials in the bladder cancer space as it pertains to the microbiome. There are some studies that are observational, so it's just gathering information on whether or not there are these differences and how can we predict these differences. But there are some interventional trials including for fecal transplants in the immunotherapy space that do include patients that have urothelial cell carcinoma. So this is a topic that we are really understanding is going to change how we think about disease. Every step of the way we learn that it's more complicated than we thought before, but I do think in the next decade or so we're going to continue to learn and find some exciting data. So with that, I want to thank you for this opportunity and welcome any questions.
So fascinating information. I don't think that there's a prescription yet to eat 2 of these every day and you'll be in better shape. I think that over the next few years, you will see more. I think that's very exciting. So I know we have a number of questions from our group, so the microphones are coming around.
I have one here, Stephanie. Here you go.
Where are you?
Left.
There you go. Okay.
You're
first
right.
Question.
Hi. Could you give a perspective about evolutionary pressure and microbiome? In other words, all of us in this room eat nothing like we did people did 200 or 300 years ago. And what kind of selection process might have played a role to where we got to today?
Yeah, I think that's a wonderful question. Certainly diet has been shown over and over to alter our microbiome, and I think that the types of nutrients and the diversity of nutrients that we eat, but also included in that, we eat bacteria. That's something that is inherent in all of our food, and so the types of foods that we eat, where we get those foods from, does change what is in our body. The question, of course, then is what do we need to have within our body? And that's the focus of a lot of ongoing work. And so I think we all know that eating healthy relates to better health. And I think there's some data out there that suggests that a healthy diet does lead to a quote-unquote better microbiome. But just remember that this is really a diverse set of bacteria, and what may be better in one aspect of health may be worse in another aspect of health as the microbiomes interplay with one another. But yeah, no, absolutely.
This may be TMI, but how is a fecal transplant done?
Great question. So there's 2 ways that a fecal transplant can be done, and there's actually an FDA-approved product for this, I believe. The most direct way, as you would imagine, is enema. But there is now a pill-coated form that can travel throughout the gut, and then when it gets to the colon is released. So some progress on that front.
Go ahead, Angie.
Okay. One of the slides talked about how the efficacy is reduced after antibiotic use, or with antibiotic use of the immunotherapy drugs that were given.
A little closer, Angie. Thank you.
Okay, sorry. So one of the slides talked about the efficacy, about the immunotherapy response after having like a BCG or another immunotherapy treatment. My question is, could that antibiotic use then also alter our own immunotherapy response, so then our own immunotherapy response won't be able to counteract cancer or whatever else, viruses, et cetera?
Yeah, that's a wonderful question. So the question is, our own bodies have some sort of immunologic response before we even talk about taking an immunotherapy, and can antibiotics change that? And I would suggest probably. I don't know that we have the data to say that, but as this data suggests that the microbiome and the bacteria that we have within ourselves train our immune system, impact our immune system, altering that may alter our own response. Now, the only thing I would add is I don't know the answer if it will make it better or worse. All I could say is that it would alter that response, because I think the microbiome is a very diverse. You could imagine that one antibiotic that kills off one set of bacteria may lead to one type of response, where another antibiotic that kills off a different subset of bacteria may lead to a different response or outcome. And so to say all antibiotics lead to one result, I think would be a bit premature. But yes, I agree absolutely that altering the microbiome with an antibiotic has the potential to change the response.
And I guess a second part of that now is a lot of us are then diagnosed with UTIs before we're diagnosed with bladder cancer. And we're giving rounds of antibiotics. Some people, I heard today, 3 rounds before they ever ended up finding out, no, it wasn't a UTI, it was bladder cancer. And women, unfortunately, I think do this a lot more often than men, because women are more prone to UTIs to begin with. And so the first thought is that. And even me, they gave me antibiotics, and in my head I knew it wasn't a UTI, but I said, well, no harm. Well, maybe there is harm, because now I'm reducing my immune response to my own cancer. I don't know. I mean, it's a thought, and it's something that, that we're all dealing with when we're taking those antibiotics and we've got cancer.
I think it's a wonderful thought. What I would say is I think it's a wonderful thought, and I think that while it has the potential to be true, I would say we're too early to have anyone leave this room thinking that is the case. I don't think we have any data at this point in time that pins antibiotic use with bladder cancer development. So I just want to make that clear. But it does lead to that thought that if we alter our microbiome, be it through medications, be it through diet, can we alter our risk for various diseases? And I think there's probably some truth to that. But I wouldn't put just for the people in the room, A equals B in that space. And I think that antibiotic use in this patient population is very common, not only in the pre-diagnostic space, but then when you talk about BCG therapy and catheterization, there's a lot of opportunities for needing an antibiotic to treat that type of thing. And so it is a very common aspect of bladder cancer, absolutely.
It was interesting looking at the different microbiomes with bladder cancer without. Has there been any further study in terms of the types of bladder cancer versus the microbiome composition, or if there's any?
Yeah, that's another wonderful question. Is do different types, different forms of bladder cancer have different microbiome signatures? And so that might suggest that, well, bladder cancer, as we know, is not really just one type of disease, but it's a big umbrella term. But different forms have very different sort of treatment paradigms and whatnot. And so I don't know of anything in that space yet. But as you can see, this paper was from 2024. I think that just leads us to an opportunity to look at that. Thanks.
I'm just looking at this slide to follow up to that question. When you look at the healthy biome on the left, has that been mapped back to diet so that you could literally say, to create this, you should do that?
So the question is, when you look at the healthy people, if this is a representation of what a non-bladder cancer comprising bladder looks like, how do we drive this signature? And I think that's the ultimate goal. I think that we are decades away from how to do that. And then the question also is it the chicken or the egg kind of in this space? So is it that the microbiome in that middle picture puts someone at risk for developing bladder cancer? Or is there something about bladder cancer itself and the signals that it gives off that may also alter that? I don't know. Certainly what the hope would be is that we could prevent disease and treat disease by altering the microbiome. And I think one of the other hopes is that instead of just looking at the millions of different types of bacteria that we have, we can then drill down and figure out what is it, which of the bacteria is causing this, and what is it about that bacteria that's causing it? Is it certain proteins and enzymes and metabolites that it's producing, so that even if we can't drive an entire population of bacteria to look like a certain thing, we might be able to understand the root cause of that, and that might be something that we could utilize to help prevent, treat disease.
So I was just going to point out, some people may not know this, that the National Institute of Health has a dedicated department, CAM, Complementary and Alternative Medicine, that I think for the last decade has been trying to legitimize both the quality and the manufacturing of alternative therapies, because billions of dollars are spent on Eastern type therapies for disease treatment. And the other, the question I have is the translational research getting into the real world in regards to urine not being sterile, and how when they do the microscopic urinalysis, culture and sensitivities, how is that translating to differentiate which is the good and the bad bacteria, fungi, and viruses?
Well, thank you for the comment, and thank you for the question. So the question is, just if I understand it, the first one is where are we at in translating some of these findings? And then the second question is, when we do a culture, I think many of you probably have had a urine culture sent and something grows from that. How do we know that that's a good bacteria or a bad bacteria? So to the first question, I think we're still very much in the early days of translating this into clinical practice. And I think a lot of the efforts, based on sort of what I said earlier, are going to be to translate into molecules that we can create and manufacture. Completely altering a microbiome becomes a challenging thing. Probably the most direct way to do that is the fecal transplant. And as you can see there, we know that one microbiome can be helpful, whereas another may not be. But when we deliver something to patients, we want to have something that's standardized and something that we can say, this worked for as many people as possible, as opposed to we got this stool from one guy and we got this stool from the other guy and we hope it works this time and not that time. So I think ultimately if we can figure out how to have a microbiome transplant that can be reproducible, not just reproducible in a one-time setting, but year after year. My microbiome probably is different today than it was a year ago, just based on what I eat and my life experiences. So I think that will be the challenge, but that will probably be also the most direct way that we can impact the efficacy of some of these drugs, by altering the microbiome. So I think that's where we'll see things in the next decade, as it pertains to a real translational product. And then as far as how do we tease out what is a good bacteria or a bad bacteria in our cultures, the way that our cultures work is to be diagnosed with a urinary tract infection, you have to have symptoms, which is an important thing. So just because you have bacteria grow in a culture does not mean you have an infection. It might just be an overgrowth of a particular bacteria, but if it's not causing you a problem, causing you symptoms, then it is not, we don't classify it as an infection. Now, could that bacteria be good or bad as it pertains to disease, bladder cancer, et cetera? I think that's what we're sort of working on. And then the other part to that question, to find all of these different types of bacteria that live within a microbiome, usually they're in minuscule amounts, and we're detecting it only because of sequencing. For someone to have a urinary tract infection, they need to have so much of that bacteria that when we dilute the urine to very dilute amounts, they grow on a dish at very high rates. And so symptoms plus high rates of growth on a dish are what we define as an infection. But being able to tease out this is a good bacteria or a bad bacteria is really the focus of the work, such as this group's, but we don't have that answer yet.
Hello. Thank you for the useful information. My name is Danny. I survived non-muscle invasive bladder cancer in 2023, and I'm in a 3-year course of BCG immunotherapy. I just finished my last maintenance this year. I got 1 more year left, and I'm completely cancer-free. I've not had a return tumor. Now, I really got into shape. I weighed over 300 pounds when I first got cancer, and like most of us, I'm sure, we decided to hit the gym, and I overdid it, but I did manage to lose about 100 to 120 pounds. And when I did that, I completely dissected everything that I ate, and I had terrible diarrhea. So I read at some point, I was going through BCG, I was past turbot, I was past a stent, and I was starting BCG. And I started to read up on it, and I saw this thing, I can't remember what it was, I think they were calling it cancer gut. And I didn't know how that affected me because it was localized to my bladder. Maybe they were talking about like colorectal cancer with this whole gut biome thing they were talking about. But I was convinced that it was something that was happening with the BCG. So I think more often than not, comorbidities is what I kind of leave these in the category of when it comes to health. It's the onion layers of what I was trying to pull apart. Diarrhea for months, I was losing weight, it was getting bad, it was painful, so I was gonna go to the doctor. Well, I started taking kombucha and doing different things like that. And it eventually turned out to be sugar alcohol because I stopped eating sugar because of the things about cancer. I'm allergic to sugar alcohol. So now that I've pulled all those onion layers away, I'm completely regular and very happy. And it doesn't seem to, at least for me, mess with my gut biome. But at the same time, I'm terribly BCG responsive. I urinate blood. I'm sick for an entire month on my maintenance doses. So I went to my doctor and I said, what can I do? He's terribly pragmatic. And he said, you can stop. This is the only option he gave me. I said, can you give me some ibuprofen, maybe something like that, to help with the pain? He says, well, that will lower your immune response. He completely refuses to do that. So I was going to quit, and I know people that are still cancer-free, that didn't finish their BCG, but I went ahead and decided, because I got 1 year left, and I'm not looking forward to those 2 months in 2026, but the proof is in the pudding, and I'm cancer-free, so I'm going to continue to go with that. But I want to always urge people to look before they jump too far ahead, start to peel those onion layers away and see actually what's going on. Sometimes it takes 2 years. I had diarrhea for almost all that time before I figured it out. So thank you so much for this information. This helps me immensely.
Well, thank you, Danny. I appreciate it, and I'm glad to hear that you've had such a good response. I'm sorry it's been a tough road otherwise.
Okay.
This might be way off topic, but is there any connection with vagal neurology and microbiome?
I don't know the answer to that question. We do know that the microbiome can generate neurotransmitters, and to that end, it may impact the vagal nerve and whatnot, but I don't know the answer.
Can you speak to the composition of bladder cancer? Like, we know that 50% of people on average have had some smoking history. What I have heard is that as people have stopped smoking, that is the composition is changing. And so I guess my question is really, is the thought behind microbiome that we may start to see a different composition of bladder cancer, younger, healthier? What are you seeing sort of in practice and as you look at the statistics? Is this going to be like colon cancer where all of a sudden we start seeing people in their 30s and 40s diagnosed more? Just curious.
That's a wonderful question. So I think we've all seen in the news and across the news that there has been quite a rise in colon cancer diagnoses for younger people, and we don't really know why that is. I think some people are speculating about microplastics or something about the diet, and it certainly would be reasonable to think that the microbiome within the gut, which we know has changed with dietary changes as well, may be a major driver in this. I personally have not pinned differences in types of bladder cancer in the younger patients, but I think that that is something that we should look at and keep a watchful eye, especially as we have seen rates of tobacco use drop precipitously in this country, but we have not seen rates of bladder cancer change. And so you would think that one would follow the other, and it has not. And I think that we really owe it to you all to try to figure out why that is. So, thank you.
Get the mic over here.
What is your thought on association of bladder cancer with PFAS or the forever chemicals? I know they've linked it with kidney and testicular cancers, but the jury is still out. It's still speculative.
Yeah, I think jury is still out. I don't know enough about that space, and I don't know if we have enough data in that area. I don't know.
Okay, probably have time for one more question. Thank you all for these great questions. And Dr. Scovell, this has been fabulous. Obviously, there are no answers. The good news is there are really intelligent people that are really thinking this through. Over the next 5 years at Beacon, we will see more change. We will see more of this research. And the good news is it will improve outcomes for patients. And it may just be the future is in fecal transplants. Who knows? But the idea is that some clever minds are working on things that we wouldn't normally think about. And science is beyond our imaginations today. And the good news is smart people are working on it. And you all have a real interest. Think about what you do eat. Think about how you engage in the environment. You know, we know that smoking is not good. And we know that people who continue to smoke have worse outcomes. And there are a lot of things we can do to try to make our lives better. But sometimes the science isn't quite there. So if it feels better for you to be changing in a good way, do those things and tell your doctors so that they know what you're doing as well. I think that's important. So I want to thank you, Dr. Scovell.
Well,
any last thoughts you wanted, impart on our group here? Obviously, they had a lot of questions.
Yeah, no, first of all, I really thank you all for being so engaged and having a lot of thoughtful questions. It was really inspiring for me, actually, to hear how interested you all are in this topic and moving the field forward. So I just want to thank you for having me in your space and really appreciate the opportunity to share this with you.
Thanks so much for joining us.